The process of nutrient digestion is completed in the small intestine. So that's the very last stop for the process of digestion. After digestion is complete, of course, we can absorb the nutrients. Water, minerals, and vitamins are three nutrients that actually don't require digestion. There's no need to break them down into smaller pieces. We already consume them in their smallest form. But the small intestine is completing the digestion of carbohydrates, proteins, and lipids. The carbohydrates are going to get broken down into usually their smallest forms of monosaccharides or maybe a disaccharide. And actually, I wanted to talk about lactose intolerance because people that are lactose intolerant can't digest the disaccharide lactose. So basically what that means is that they don't have an enzyme, and basically this enzyme is needed to break down the lactose. The enzyme is called lactase. So people that are lactose intolerant, if they consume a lot of lactose, what that means is the lactose comes in one end and it goes right out the other end because they can't break it down or absorb it. So often the symptom of consuming lactose by the lactose intolerant is diarrhea. The proteins get digested usually into amino acid form, though some are actually absorbed as small peptides. And lipids are broken down a little bit. It just kind of depends upon which lipid we're consuming. Most of the lipids in our diet are triglycerides, and the triglycerides are digested into fatty acids and monoglycerides. Cholesterol, though, generally is taken in and absorbed in the same form. As I said, most nutrients are absorbed in their smallest form or maybe a slightly larger version of their smallest form. In order for complete absorption to occur, we must get the nutrients out of the lumen of the small intestine and basically into the cells of the body. But we have to do more than just that even. We have to actually get those nutrients out of the cells of the body and into circulation so that they can be used by other cells in the body. Essentially what's going to happen is we're going to take the nutrient out of the small intestine lumen, move it into a small intestine cell, move it out of the small intestine cell and into the general tissue fluid or interstitial fluid. Then we have to get the nutrient out of the interstitial fluid and into either blood or lymph in order to make sure it gets into circulation. This drawing shows the major players involved in the process of absorption. We have to get the nutrients out of the lumen of the small intestine into the small intestinal cell, out of the small intestinal cell into the interstitial space, and then out of the interstitial space into either a capillary or lacteal. Don't forget that the cells that are in contact with the lumen are simple columnar epithelial cells. They're part of the epithelium of the mucosa. Sometimes we call these cells enterocytes or brush border cells. I have them labeled as brush border cells on here. So when we absorb our nutrients, we have to get the nutrient across two cell membranes. We have to get the nutrient across the cell membrane that's facing the lumen and the cell membrane facing the interstitial space. This is the apex of our epithelium, so we'll call this the apical cell membrane. This is going to be the base of our epithelium, so we'll call this the basal cell membrane. Now remember, cell membranes are made of lipids. They're phospholipid bilayers. So in order to get things to cross the phospholipid bilayer, it's a little complicated, right? So we're going to take a look at those processes. So as I just said, the nutrients have to cross two cell membranes to get into the interstitial fluid. They have to cross that apical cell membrane and the basal cell membrane. So what we need to do is go back in our brains and go back to our cell biology and think about, how do substances cross cell membranes? What were those mechanisms that we learned about that explained how substances could cross that phospholipid bilayer of the cell membrane? So this is our review for transport of substances across the cell membrane. We have four basic mechanisms we can use to transport things across the cell membrane: simple diffusion, facilitated diffusion, active transport, and then bulk transport. And we will use all of these in order to get nutrients across the cell membrane of those brush border cells. But let's just quickly review each of these different types of transport. With simple diffusion, we're transporting the nutrient right across the phospholipid bilayer, not through a protein, but actually across the phospholipids. And this is going to be driven by a concentration gradient, meaning the nutrient will travel from an area of high concentration to the area of low concentration. Facilitated diffusion is similar in that it is being driven by a concentration gradient. However, the transport is not happening through the phospholipid bilayer. The transport is going through a protein, a facilitator that's located in the phospholipid bilayer. With active transport is similar to facilitated diffusion in that we are using a protein transporter in the cell membrane, but in this case, the substance is not being driven by concentration gradient. Instead, it's moving against its concentration gradient, meaning the nutrient is moving from an area of low concentration towards the area of high concentration. Because we're trying to defy physics here, we have to supply energy to drive active transport, which means we need ATP. We either need ATP directly or indirectly. So as I have on the slide here, sometimes we use ATP to create a concentration gradient for a substance. Let's call that substance A. Then we use that concentration gradient to transport another substance, we'll call that substance B, across the cell membrane. Basically what I'm saying here is that we have a concentration gradient for substance A. So substance A is capable of facilitated diffusion across the cell membrane. And we're going to couple that with another substance, substance B. Basically, substance B is riding on the coattails of substance A. So whenever substance A is moving, substance B will move too. And this is a type of active transport where we're going to be using ATP to set up that initial concentration gradient for substance A. But then when we transport substance B, that part doesn't require ATP because it's using that facilitated diffusion along with A to get into the cell. So this is a type of active transport, but I just want to make you aware of it in case you read about it or something. I'm not going to talk about this in a lot of detail though. Anyways, the other transport mechanism is bulk transport, also known as vesicular transport, where we move large amounts of substance across the cell membrane. And of course, this requires the formation of vesicles, hence why it's called vesicular transport. So which nutrients get absorbed in which way? Simple diffusion is very special in that only certain things can simply diffuse across the cell membrane. Gases, water, and anything hydrophobic, such as fatty acids and fat-soluble vitamins, are the things that can cross the cell membrane via simple diffusion. So we're not really worried about gases here because that's something for the respiratory system, not the digestive system. But water and hydrophobic nutrients are going to cross the cell membrane via simple diffusion. So cholesterol, fatty acids, monoglycerides, fat-soluble vitamins, all of those things can cross via simple diffusion. Several nutrients will cross via facilitated diffusion. Actually, all of the nutrients are capable of crossing via facilitated diffusion except for the hydrophobic nutrients because, of course, they can simply diffuse, and why would you bother to add a protein to transport that substance if it already can be transported without the protein? I wanted to make note of the fact that water, of course, is also capable of facilitated diffusion. The type of protein transporter that's used to transport water is called an aquaparin. But keep in mind that we usually don't refer to water as being transported via facilitated diffusion. Instead, we call it osmosis. So osmosis is the diffusion of water across the cell membrane, and osmosis includes both facilitated diffusion of water and the simple diffusion of water. There are a few nutrients that will be transported via active transport, and these include carbohydrates, amino acids, nucleic acids, and some of the minerals. And then, of course, there are a few nutrients that will be transported via bulk transport. Vitamin B12 is one of them, and we kind of learned about the special circumstance with vitamin B12 in a previous unit. Some nutrients will cross the cell membrane via bulk transport. One of them is vitamin B12. If you recall, vitamin B12 requires intrinsic factor in order for it to be properly absorbed. Intrinsic factor is released by the parietal cells of the stomach. Intrinsic factor will combine with vitamin B12, and that will allow endocytosis of the vitamin B12. So that would be bulk transport into the cell for that vitamin B12. Fatty acids can also come in this way. We'll talk about that in more detail later. And fat-soluble vitamins do pretty much whatever fatty acids do too. This slide is essentially a summary of the different types of transport mechanisms that we use in order to absorb the various nutrients. And I've included the transport mechanism across the apical cell membrane as well as the transport across the basal cell membrane. For example, with lipids, to cross the apical cell membrane, they will use simple diffusion, but to cross the basal cell membrane, they will use exocytosis. A couple things that I wanted to mention. One is I wanted to mention iron. So iron is a mineral, and because women have menstruation and they lose more blood than men do, women actually have 4 times as many protein transporters for iron than men do. Calcium, which is also a mineral, can have differences in absorption too. In this case, it's really dependent upon a hormone, calcitriol, also known as vitamin D. So calcitriol is stored in the kidneys, and it gets released when our blood calcium levels are low under the influence of parathyroid hormone. And the hormone calcitriol travels to the small intestines, and basically it tells those brush border cells to make more protein transporters for calcium. In this picture, what we have is basically a summary of that table. So it's the table in pictorial format. Now, what this is saying is that monosaccharides, amino acids, and nucleotides will be transported across the apical cell membrane via active transport, but then they are transported across the basal cell membrane via facilitated diffusion. Or with the case of water-soluble vitamins and minerals, those will be crossing the apical cell membrane via facilitated diffusion or active transport, and likewise they will cross the basal cell membrane via facilitated diffusion or active transport. Osmosis is not shown on here, but osmosis is being transported via osmosis all over the place. So now that we have the nutrients into the interstitial fluid, we have to move the nutrients into either blood or lymph. Don't forget that lymph essentially will transport substances to the blood. So ultimately, it's all going to end up in the blood. Everything except for the hydrophobic nutrients will be put into the blood capillary of the mucosa of the small intestine. And basically, this happens via the nutrients entering the capillary through those intercellular clefts. So in the picture here, these yellow oblong things are supposed to be the cells of the endothelium of the capillary. And notice that there are spaces between them. Those are the intercellular clefts. Those red things are supposed to be red blood cells. So the nutrients just travel through those intercellular clefts in order to enter the capillary. This is actually something we learned about back in the circulatory system. We call this capillary reabsorption. Eventually, the blood from the small intestines is delivered to the liver. I shouldn't say eventually, because actually it's pretty direct. The blood from the small intestines is directly sent over to the liver via a special vein called the hepatic portal vein. And this is important because the liver has the job of detoxifying any toxins that we might have absorbed with our food. And we definitely absorb a ton of toxins with our food, so this is a really important process. The hydrophobic nutrients, though, are not going to enter the capillaries. Instead, they're going to enter lacteals. Lacteals are just a specialized lymph capillary. And don't forget that the lymphatic system, like I said, is eventually going to deliver this stuff to the circulatory system anyway. For a visual representation of the lacteal taking in the hydrophobic substance, it kind of looks like this. So the cells that make up the wall of the lacteal are kind of like flaps, and when the flaps open, the hydrophobic substances will enter. I've been kind of saying that there's this special case with lipids, so I want to address that now. We know that bile aids in the digestion of lipids because it's an emulsifier that allows the lipid to mix in the watery environment where the lipases are, and we need those lipases to break down the lipids. Micelles are basically globules of fat and bile. And the micelles, they're floating around in the food, in the small intestine, and they pick up digested lipids and the lipid-soluble vitamins and transport those to the apical cell membrane of our brush border cell. The lipids are then going to enter the cell via simple diffusion. Once the lipids are inside the cell, and I keep saying lipids, but it also includes the lipid-soluble vitamins. Once the lipids are inside the cell, they're packaged into structures called chylomicrons. Chylomicrons are very similar to a micelle, but instead of globules of fat and bile, we have globules of fat and protein. There's an outer shell of protein that is basically encasing the fat. The chylomicrons are then sent to the basal cell membrane and released into the interstitial fluid via exocytosis. Now that the chylomicrons are in the interstitial fluid, they will be picked up by the lacteals and eventually sent to the blood. The reason why the chylomicrons have to enter via lacteals is because they're actually too big to get through the intercellular clefts of a typical blood capillary. The little flappy openings on the lacteals are much bigger, and of course that makes it easier for the chylomicrons to enter those as opposed to the capillary. The chylomicrons are eventually repackaged with more fats, and this happens in the liver, and it forms something called LDL, which stands for low density lipoproteins. The LDL is then sent out into circulation, and basically the job of LDL is to deliver fats throughout the body. Whatever organs need the fats, right? Heart, muscle, whatever it might be, the LDL is delivering fats throughout the body. LDL is eventually converted into something called VLDL, which is very low density lipoproteins. VLDL, like LDL, is delivering fats throughout the body. Cholesterol is a part of the fats that are in these lipoproteins. We refer to LDL and VLDL as the bad cholesterol, and then HDL is the good cholesterol. So let me describe HDL, and I'll talk about why one's good and why one's bad. Actually, you'll probably figure it out. HDL stands for high density lipoprotein, and its job is to pick up extra fat throughout the body for disposal. So you probably just had a light bulb go off. You're like, oh, the HDL is getting rid of fat from the bloodstream, whereas LDL and VLDL is spreading fat throughout the bloodstream. And of course, fats like cholesterol increase our risk for atherosclerosis and other cardiovascular disorders, right? HDL is the healthy cholesterol because it's removing cholesterol from the body. So higher HDL levels are good, and lower LDL and VLD levels are good.